All right, hello everyone. Let's go ahead and get right into this. This is Lab 9, Day 1 of a two-part lab that focuses on detecting if a particular food source was derived from a GMO plant. Okay, so you can see there that the pig right here is an example of what we call a genetically modified organism. If you look closely, notice how the snout here, as well as the hooves, have a green tint to them as compared to the other pig or hog, if you want to call it, that's back there. So the objectives of this lab are what we hope you'll achieve or be able to explain by the time you complete the lab is be able to define the term genetically modified organism or GMO, describe some of the benefits and controversies regarding genetically modified organisms, describe a technique, a lab technique called PCR, which stands for polymerase chain reaction. What this is DNA replication in vitro or in a test tube, and then describe the roles of two control foods and the functions of the two different primer sets in this experiment. And basically, what this has to do with these controls is to ensure that the technique PCR works properly, and that if you get a positive result for a test food, as far as the test food was derived from a GMO plant source, you want to know that you're not getting a false positive, that it's actually positive. So what is a genetically modified organism? This is an organism with DNA that has been deliberately altered. Okay, very often the DNA of a GMO is altered by adding DNA from another organism. We call this a transformation. Now, the way we used to do it when I worked in the lab is we would call it a transformation if it was a bacterial cell that we were adding foreign DNA to. Okay, if it was a eukaryotic organism or eukaryotic cells, we would use the term transfection, or we would say transgenic down here. But if you go up one line, it says the donated DNA usually includes one or more genes that encode for proteins not naturally produced by the recipient organism. This is something we'll cover in Chapter 17 to help clear up some of the confusion that will occur with this lab. Now, because DNA is transferred from one organism to another, GMOs are sometimes called transgenic organisms. Again, this primarily is for multicellular eukaryotic organisms or animals. Now, genetic modification is meant to enhance an organism, often by accelerating its growth or improving its disease resistance. So again, here you can see there are mice. This one here is a GMO. This one is not. This one here, as well as the pig or the hog on the first slide, actually are both expressing that protein that we focused on for Lab 8, which is green fluorescent protein. But you can see here, not only is this mouse expressing this protein, but it's only expressing it in certain regions of its body, such as the ears, around the eyes, the feet, and the tail. This here, I'll just let you look at here. It just goes over some plants there and food sources or plant and animal food sources that have to do with GMOs. Now, there are a number of genes that may be transferred to genetically modify an organism. I'm going to jump down to the second one. This first one talks about Roundup, and that became a big issue in court as far as causing cancer. But I'm going to jump down here because this is more pertinent to what we're leading into. But there's a European corn borer insect, okay, that's a pest that feeds on corn plants, right? So some corn plants have been modified with the gene from a bacterium called Bacterium genesis. So now we're talking about corn plants, which are eukaryotic multicellular organisms, and over here we're talking about a unicellular prokaryotic organism, okay? And what has happened is that for this type of bacterium, Bacillus thuringiensis, okay, it was found to express a protein that functions as an endotoxin. In other words, it poisons insects such as a corn borer larva. Whenever, what we call these insects would consume or ingest bacteria unknowingly. So what they did is they took the gene that coded for this protein endotoxin, they isolated it from this organism, okay, and they used it to transform the cells of the corn plant, so that now if the corn borer larva fed on these GMO corn plants, they would be consuming that toxin that originated from the bacteria, but now that the corn plant, which is a GMO, could now express itself. Therefore, instead of having to use pesticides or insecticides on the plants themselves are expressing this bacterial endotoxin, which kills the larva, so the larva is able to feed a little bit on the plant, but not completely. But they haven't just used this toxin from Bacillus thuringiensis to transform corn plants. They've also used it to transform peanut plants. So if you look here, they're showing you, here's an organic peanut plant. It's, in other words, we're not just talking about insecticides haven't been used on it, but it hasn't been transformed. It's not a GMO plant source. When it's exposed to these corn borer larvae, they feed freely on the plant leaves, the peanut plant leaves. However, these leaves here, okay, are the leaves of GMO peanut plants. And you can see the corn borer larva here is dead. Now you can see a little bit of the plant leaf has been consumed, but as the insect is consuming the plant leaf tissue, it's consuming the endotoxin, which kills it. So these right here, these GMO peanut plants, they don't have to be treated with pesticides or insecticides, okay, because they've been transformed to protect themselves. They've been enhanced. Now, making a genetically modified organism, the first step, okay, in the genetic modification is to identify a protein that has the potential to improve another organism. So I left out the word another, okay? So that's the first thing they want to do. Identify a protein that has the potential to improve another organism. Okay, so what they'll do is they'll look at one organism, such as the bacterium B. thuringiensis. When they discovered that protein endotoxin, okay, they were able to eventually track down the genetic information that coded for that protein, and they would use it to improve or modify corn plants, thus improving those corn plants. Now, as I said before, once the protein is identified, the gene for that protein is isolated from the DNA of the donor organism. So for these first two lines here, I'm going to go to the next slide. That's this stretch right here, all this here. Okay. First, they have to identify a protein expressed by a certain organism that scientists feel would be beneficial to a different species, that could enhance another species. Most often it has to do with proteins expressed by bacteria that kill insects, in other words, proteins that function as endotoxins. Scientists first started focusing on those. Okay, and once they identified the endotoxin as being a protein, they know that in any living organism, okay, that expresses proteins, the information on how to make those proteins can be found in the living organism's DNA, in its genetic information. So if you look down here, all living organisms have their genetic information stored in the form of DNA. So scientists knew this also. So therefore, once they identify a bacterium such as Bacillus thuringiensis was able to produce an endotoxin that is a protein, they were able to, in a manner of speaking, backtrack, okay, and locate the genetic information stored in the bacterium's DNA, okay. And when they scanned the DNA, they would scan the DNA until they would were able to identify a particular region of that bacterial DNA that formed a gene that coded for that protein toxin. Okay. Once they identified that region of DNA that formed a gene that coded for that protein endotoxin, then what the scientists would do, okay, is they would extract DNA from many bacteria, and then they would cut out and isolate the gene. Okay, so this is where they've taken it up to here. Now, when they do this, on the other end, they already know what other organs, what other different species they want to transform into a GMO. They want to enhance its characteristics, plants being a key source. Okay, so this is what it's talking about up to here. Up to this point, okay, the gene that codes for that protein will be isolated. Now I'm going to skip this. This is more technical stuff that we don't really need right now. What I'm going to do is I'm going to jump ahead. Okay. Now the catch is this, okay? Before the gene, sometimes it's a clone gene, but before the gene can be introduced into the recipient organism, and like a corn plant, okay, it has to be modified so that it will be properly expressed in those recipient cells. What they're getting at is this. This right here, okay, is representing this right here. And these two right here are representing the gene that comes from the bacteria. Okay, so everything's circled that I put there. So we have bacterial DNA. There's a region of that DNA that forms a gene, which has been extracted and isolated, that codes for the protein endotoxin. And what scientists discovered is that if they just take the gene alone that codes for the protein and insert it into an embryonic plant cell, okay, the plant cell will not recognize that gene. Even though all living organisms have their genetic information stored in the form of DNA, scientists discovered it's not enough, okay, to just simply insert the gene. What they discovered, okay, is that genes need to be bracketed by DNA sequences that we call DNA regulatory sequences. Okay? There are two key types of DNA regulatory sequences. One is called a promoter, and the other one is called a terminator. Okay? Regulatory meaning control. Okay? These two sequences help with controlling or help with control of the gene being expressed by the recipient, by the organism or the cells we want to transform or convert into a GMO. Now the promoter is placed upstream. Okay, the terminator will be placed downstream. So on this figure here, okay, this stretch right here, what they're showing you here is this stretch would represent the promoter and this stretch would represent the terminator. The gene itself is here in the middle. Okay. So they show you the color-coded DNA changes. All right. Now, the reason for this is because the protein enzyme responsible for expressing this gene so that eventually the protein endotoxin will be produced, it needs help locating the gene. That's where the promoter comes into play. And when that protein enzyme has a promoter, a DNA regulatory sequence called a promoter, to help it find the gene, okay, that enzyme will bind to the promoter, slide over, and start to express the gene so that eventually the protein is made by the cell. But when it starts to express that gene, okay, the promoter helps it locate it and know where to start expressing the gene. The terminator helps that protein enzyme know when to stop. In other words, hey, this is the information right here. Don't go beyond it. Don't start before it. That's what the purpose of these two regulatory sequences are, so that the gene is expressed properly to produce a functional protein. Well, what scientists discovered, okay, is that when they looked at other living organisms and when they looked at viruses, which are considered not alive or they're not dead either, what they found is that there are a variety of different promoters in different species and viruses, and there are a variety of different terminators in different species and viruses. So through a lot of trial and error, scientists eventually discovered that a promoter that works very well in helping create a genetically modified organism is a promoter known as a 35S ribosomal gene promoter. Okay, except this promoter more popularly goes by simply CAMV promoter. This was a promoter found attached to genes in a virus called the cauliflower mosaic virus, thus the CAMV promoter. They found that even though this is a viral promoter, this DNA regulatory sequence works very well when attached to prokaryotic and eukaryotic genes when wanting to create a genetically modified organism. Okay? On the other end, they found that in the bacterium, Agrobacterium tumefaciens, excuse if I didn't pronounce that correctly, okay, there is a terminator known as the nopalin synthase gene terminator, but it more popularly goes by the NOS terminator. Okay, they found that this terminator works very, very well when you put it downstream, okay, or at the end of the gene. So upstream means at the beginning of the gene, downstream means at the end of the gene. In other words, it's like if you were looking at it, okay, upstream would be to our left, downstream would be to our right as far as the gene goes. And this is terminator, okay, and it's been found. Okay, now this is from a bacterium, this is from a virus, and then the gene that they want to use to transform an organism transform it into a GMO? Well, it could come from a variety of different species, but they found these combinations work. Well, eventually what they discovered, or that the general population discovered, okay, for those that were interested, is that this CAMB promoter and this NOS terminator, they are used the majority of the time as DNA regulatory sequences that are attached to a gene that a scientist wants to use to create a GMO, especially for GMO plants. Therefore, if somebody wanted to know, are those plants GMOs, but they don't trust the person that's giving them an answer, if they can obtain some of that plant tissue and they have access to the necessary lab equipment, all they would have to do is try to detect if this promoter or if this NOS terminator is present in the plant DNA, and then they would be able to verify, hey, look, that's not a naturally occurring plant. It's not organic at all. It is a GMO. So then what will happen is once they add these regulatory sequences to the gene, they will then insert that DNA into a plant embryonic cell, and then that cell will divide it into many cells, each cell inheriting this foreign DNA, this foreign gene from the bacterium, till eventually we have a full-grown plant. With every plant cell, okay, inheriting that gene that came from the bacterium, therefore every plant cell has the ability now to produce that protein endotoxin. And when it comes to plants, plants not only can sexually reproduce, but they can asexually reproduce. So if you allow this plant to asexually reproduce or self-fertilize, it'll produce offspring that will inherit that same foreign bacterial gene. Therefore, you don't have to go through this process again. This one plant will reproduce, producing many plants, and each of its offspring themselves can asexually reproduce, producing many more plants, till you have a large crop of plants that are all GMOs producing the same bacterial toxin. This here is to show you the enzyme that is responsible for catalyzing gene expression, okay, helping a cell, okay, read the genetic information stored in its DNA to make the necessary protein it needs to do a specific type of work. Well, this enzyme here, RNA polymerase, this is a protein enzyme that needs a promoter to locate the gene, but also needs a terminator to know when the gene ends. And we'll talk about this more in chapter 17. Okay, the lab technique that we use, okay, and that is actually used in research labs to determine or to detect if, okay, an organism is a GMO, that lab technique is PCR. PCR stands for polymerase chain reaction. PCR is simply DNA replication in vitro in a test tube. So if you look here, PCR is a molecular biology technique used to make identical copies of a specific DNA sequence. So it's DNA replication. This is chapter 16. Okay. Now, some differences between PCR, which is DNA replication in vitro, and DNA replication in a living cell, which we say in vivo. Okay, well, one, PCR is in a test tube in vitro versus a DNA replication in a living cell, which is in vivo. PCR, okay, with PCR, you determine what you want to replicate. So PCR copies specific target DNA sequences, unlike in a living cell where DNA replication copies all of the DNA in that cell. PCR uses different temperatures, okay, instead of different proteins or enzymes to carry out the steps of DNA replication, okay. However, what they have in common for both DNA replication in vivo and DNA replication through PCR in vitro is that we always need an enzyme called DNA polymerase, and I'll talk about this more in a second. Okay. Now another difference is in a living cell when DNA replication occurs. Okay. This enzyme DNA polymerase, in order to locate the DNA that it needs to replicate, it needs the help of short single-stranded pieces of RNA. We call these RNA primers. But what scientists discovered is that this enzyme DNA polymerase, it's not picky, okay? A short single-stranded piece of RNA called an RNA primer or a short single-stranded piece of DNA called a DNA primer will do just as well to help DNA polymerase locate the target. So when you perform PCR, everybody, the key to the PCR working is in the primers here, okay? You can isolate the DNA of interest that you want to determine if it contains, okay, foreign DNA. So if we extract DNA from a plant and we want to know is it a GMO plant or is it organic, okay, whatchamacallit, that's all. But the only way we're actually going to be able to determine whether or not that plant is a GMO is through these primers. These primers, normally the way it works is scientists design these primers to locate only GMO DNA. Okay, so it's in the primers that allow you to detect, okay, if your plant or if a food source, okay, is from a GMO plant. So as I said, in vivo DNA replication requires RNA primers, short single-stranded RNAs, which is depicted here. But for PCR, it's different. Now, what I'm going to do is I'll talk about the primers a little more in a second, but what I want to go over overall is what we normally do is we give you these small little test tubes called Eppendorf tubes or PCR tubes. And to these PCR tubes, you add 5 components. The 5 components are your template DNA. Listen carefully. In today's lab, okay, you have 3 different template DNAs. Okay? You do not put all 3 of these template DNAs into the same tube. So at a minimum, you will have to set up at 3 PCR test tubes. Each tube will be containing one of those 3 template DNAs. The template DNA is your target DNA. Okay, it's your DNA of interest, okay, that you're looking to detect and replicate something of. This has to be added to these little PCR tubes. The second component, and the DNA is in solution, it's usually in a buffer. The second component you would add to that little PCR tube are your primers, except these are DNA primers. They're short single-stranded sequences made up of roughly 20 DNA nucleotides, so we call them short-stranded, single-stranded DNA sequences, okay? And these DNA primers, as we call them, okay, they are what are going to recognize your target DNA sequence. And when they bind to that target sequence, they don't bind anywhere to the target sequence. They bind to the target sequence to where they flank the sequence. So if you look here, they show you primer A, primer B, okay? And they're showing you one of the primers binds to one end of our target, and the other primer binds to the other end of our target. If you look up here, this is your target DNA, double-stranded DNA, and now you can see how the primers are binding to one end or the other. And then our target DNA is all of this here. These primers, okay, are responsible for recognizing your target sequence, your target DNA. Okay, but they have to bind to where you're able to make copies of that target DNA. Make a large quantity of copies, therefore making it easier to see your DNA, okay, through a different type of technique that we'll talk about next week to verify or determine if your food source is from a GMO. Now, these primers are also in solution in a buffer. Another component that you add to this little PCR tube is an enzyme called DNA polymerase, but the type of DNA polymerase that is used was isolated from a prokaryote of the domain Archaea, Thermophilus aquaticus. This unicellular prokaryotic organism was found to live in hot springs, live at very high temperatures, as high as it can survive as high as 90 plus degrees Celsius. They call this Thermophilus aquaticus DNA polymerase, Taq polymerase. Obviously, the T is for Thermophilus, the AQ is for Aquaticus polymerase. Most people will say Taq DNA polymerase. Okay, this enzyme is also in a solution in a buffer. This is a protein enzyme, and it is this enzyme that needs the help of the primer to replicate or copy the target sequence. Now, the fourth component that's in solution that's also added to the PCR tube is a solution containing a mixture of the 4 different types of DNA nucleotides. Okay, because it's not enough for DNA polymerase to have a primer attached to the target sequence. In order for DNA polymerase to make a copy or replicate that target sequence, it needs DNA nucleotides that it can covalently bond together. Okay. The last component that's added to this little PCR tube is a buffer solution. This buffer solution has 2 key parts to it. Okay. This buffer solution is a solution that has a certain pH to make sure the enzyme is happy so that it can do its job. But the buffer has a second component. This second component is a cofactor. The cofactor are magnesium ions. This Taq or DNA polymerase, it requires magnesium ions as cofactors for it to be able to function properly and thus recognize the primer and replicate the target. So you would have added these to your test tubes, and in actuality what happens is these bottom 4 would actually be given to you as what we call a master mix. And then what you would do is transfer a solution called a master mix containing these 4 components to a small bar tube, and then you would transfer one of your 3 template DNAs to that tube. Now, once you have all 5 components in a PCR tube, okay, and I put these a little out of order. I'm going to jump out of order here because I got ahead. What you would do next is take these little PCR tubes, okay, with the 5 components, and you would take them to an instrument called a thermocycler, and you would place them in the thermocycler. Now, the thermocycler would be programmed, okay, it would be programmed to take each of those 5 components in your PCR tubes, or to take your PCR reactions as we call them, okay, take those 5 components making up your PCR reactions in PCR tubes, take them through 3 temperatures. So PCR not only requires 5 components in solution that you add to a small PCR test tube, okay, but then you want to take those 5 components through 3 temperature steps. And you're not going to take them through these 3 temperature steps one time. You're going to take them through these 3 temperature steps 30 to 40 times, or what we refer to as 30 to 40 cycles, sometimes just referred to as a PCR cycle. Okay, so what will happen is all 5 components in the PCR tube will first be exposed to a temperature, okay, a very high temperature of approximately 94 degrees Celsius that we call the denaturing step. Okay, the denaturing step, what it does is it takes our double-stranded DNA, okay, and it causes our double-stranded DNA, our 2 strands of DNA, to detach. Or in other words, it causes our double-stranded DNA to turn into 2 separate single strands of DNA. That's the denaturation. In other words, the denaturation step is breaking the hydrogen bonds holding the 2 DNA strands together. So we go from double-stranded DNA to 2 separate single strands of DNA. Then what will happen is the temperature will drop down from about 94 degrees Celsius for the denaturing step down to approximately 59 degrees Celsius for what we call the annealing step. Okay, the term annealing comes from the word anneal. Okay, and what they mean by anneal is that the short single-stranded DNA primers, okay, will attach to the single strands of DNA. Now the primers come in sets. Okay, that way one primer will bind to one of the single strands of DNA, and then another primer will bind to the other strand of DNA, and they'll be at opposite ends of the 2 single strands of DNA that are really made up your target DNA. So in other words, the primers are binding to your target DNA sequences that are now part of two separate single strands. Okay, so the anneal just means the primer is attached to your target sequence. Then what will happen next is the temperature will change. The temperature will go up to approximately 72 degrees Celsius because this is the temperature that Taq DNA polymerase, or this is a temperature that is optimal for Taq DNA polymerase to now extend or replicate, okay, make a copy of these two single strands of target DNA. The Taq DNA polymerase, there's many of them, okay, there's not one, but there's a group of them. And one will grab to the primer on this target single strand, and one will grab onto the primer here on this target single strand. And what they will do then is replicate, in which they're showing you here in pink. Now, if you look at the figure here, they're actually showing you 3 cycles. So it shows you, we begin with denaturation, we go to anneal, and then we go to extend. That's 1 cycle. Then it says repeat. We go to denaturing again, we go to annealing again, we go to extend. That's the 2nd cycle. And then it says repeat again. Denature, anneal, extend. So on this figure alone, they're showing you 3 of 30 to 40 cycles. Normally, when we have y'all do it, we have the PCR reactions go through roughly 30-35 cycles. So that's what this figure here is talking about. Now notice, all 3 of these temps, denaturing step is carried out at 94 degrees Celsius, annealing step is carried out at 59 degrees Celsius, and the extension step carried out at 72 degrees Celsius. Notice all 3 of these temps are way above our body temp, 37 degrees Celsius. Okay, so again, there are 5 components that you put into a small PCR tube. Once you have these 5 components highlighted in blue added to a PCR tube in solution, then you'll place that PCR tube in a thermocycler here, and that thermocycler is programmed to take those 5 components in your PCR tube and expose them to these 3 different temperatures. Okay, each of these temperatures is given a name. Denaturing step, that's what 94 degrees is. 59 degrees is called the annealing step. 72 degrees is called the extension step, and it tells you here what's going on when it happens. You will be asked questions, okay, in pre-lab and post-lab about these 3 steps. Make sure you give me complete answers and temps. In the post-lab, you will be asked questions about these 5 components. Make sure you give me complete answers, but then it's going to ask you, what if one of these 4 components was missing? Okay, if one of these 4 components was missing, which of these 3 steps could not occur? Case in point, if you add these 4 components but forget to add the template DNA to your PCR tube, well, you can't even get the first step done, because if you don't add the template DNA, you don't have any target DNA to denature. So the first step doesn't happen. Or another way to say it, none of the steps happen. Okay. If you forget to add, say, DNA polymerase, but you add the other 4, well, if you forget to add DNA polymerase, if you look here, DNA polymerase comes in step 3. Step 1 and 2 can happen, but step 3 won't happen. So make sure you read that question carefully in your post lab and give me a complete answer. I'll let you read that about troubles. Now this part has to do with your lab also, everybody, and you'll see this at the beginning of the lab procedures. Okay, and you'll want to write this in. The beginning of the lab procedure, there's space there where it talks about template DNA. There's a total of 3 template DNAs, but 2 of them serve as controls. One is a negative control, one is a positive control. Okay, the negative control is non-GMO plant DNA that was extracted from oatmeal. Okay, the other control DNA, template DNA, serves as your positive control DNA. Okay, this is plant DNA that also contains GMO DNA, and it's been extracted from plants called soybeans. So listen carefully. Samples 1 and 2 have plant DNA. The only difference between these two is that sample 2, serves as template DNA that also contains GMO DNA. Okay, they both have plant DNA, but this second sample has a little extra DNA called GMO DNA. Then what you would do, normally we would give you, y'all would work in 6 groups, and we would give you 6 different test foods. Could be anything from Doritos, Cheetos, to Fruit Loop cereals, to papaya, to veggie sausage, to cornmeal, and each group would go up and choose one of these test foods. Okay, and then your job would be to extract DNA from the test food, isolate DNA, and then perform a PCR reaction on that test food to determine, okay, if that test food is from a GMO plant source or if that test food was derived from a non-GMO plant source. Okay, that's the reason for using these other two. Okay, the non-GMO plant DNA from oatmeal, it serves as a negative control, negative meaning it doesn't have any GMO DNA. The GMO plant DNA extracted from soybean, well, it has plant DNA, but it also has GMO DNA. So we use positive to say this is positive for GMO. Your test food, we don't know. Okay, that's something you're going to determine through PCR and DNA gel analysis. Now, as I said, it's not enough to have your target or template DNA. You also have to have primers. And what would actually happen is each of these 3 templates, each one, okay, you would use for 2 separate PCR reactions for a total of 6 PCR reactions. Okay, now why would you use each template DNA for 2 separate PCR reactions? It's because in one PCR tube, you will not only take one of these 3 template DNAs and add it, but you will then add what we call plant primers. To a second tube, you would take the same template DNA and add it to a second PCR tube, but now you would add a different primer called the GMO primer. Okay. Now, why would you want to take template DNA, such as non-GMO DNA, and put some of that in one tube with plant primer, and then take some more non-GMO DNA, put it in a separate PCR tube, and add GMO primer? Well, it has to do with this. The first primers that you're going to use, plant primers, these serve as a control for the PCR reaction. Okay? Listen carefully. All 3 of your template DNAs, at a minimum, they have plant DNA that contains chloroplast DNA or chloroplast sequence. All 3 of these should have it. Therefore, the reason for using these plant primers, okay, is to ensure that your PCR reactions work. You don't want to get a false positive or a false negative, not realizing the PCR reaction didn't work. Maybe something went wrong with the thermocycler, okay? Maybe it had a glitch in the program, or maybe you did forget to add something to one of your PCR tubes. Okay, you want to make sure you're not getting any false negatives or false positives. So one thing you want to make sure is that the DNA, all 3 template DNAs, you want to make sure that they are functional and that they can be used for a PCR reaction. But in the case of your test food, there's a second reason for using these plant primers. Sometimes what happens, especially when it's papaya, is students actually are not successful in isolating DNA from the papaya. Therefore, when they run their PCR reactions, okay, for GMO, the papaya comes out negative, but it's a false negative. In other words, papaya sometimes is actually from a GMO plant. But the problem is they didn't extract DNA successfully, and they didn't know it. Okay, so this is the purpose for these plant primers. You should get a positive result using these plant primers when combining them with all 3 different types of DNA. You should always get a positive PCR result. Okay, DNA should be replicated. So this is a control. The second PCR reaction, or the reason why you would set up a second PCR reaction, okay, is to determine if the food source was derived from a GMO plant. In other words, the purpose of the lab. So you would take this non-GMO DNA, okay, and you would add it to a tube, and then you would add GMO primer to that tube, as well as the other three components, okay? The GMO primers, there's actually two types of GMO primers. Some of the GMO primers recognize that CAMV promoter. Some of them recognize the NOS terminator sequence because it's been found that one or both of these regulatory sequences are often used to create a GMO plant source. And what they also found is that these two sequences overall are part of GMO DNA. And when a GMO plant is used to make a food such as Doritos, okay, that GMO plant DNA actually gets carried over to the food. And there were those scientists that discovered that companies may want to tell me that this food was derived from a GMO plant, but I don't need you to, because I've discovered that the DNA from the GMO plant, when you're using it to make food such as Doritos, that GMO DNA, as well as all plant DNA, gets carried over into the food. So now I can just extract it from the food and I can run a PCR reaction using GMO primers that will detect some of that GMO DNA, specifically CAMV promoter and the NOS terminator. Okay, so for each template DNA, you will set up two PCR reactions, one with plant primer and a second one with GMO. Okay. So for your lab report, you make sure you answer your pre-lab questions and make sure you complete all 4 questions in the post-lab. Okay, make sure your answers are complete. Okay, everybody, make sure you give me complete answers. Some of the questions you're being asked are also to help you with Lab 9, Day 2. Y'all take care now. Bye.